Next speaker is Ms. Sahar Salehi from Tohoku University. The title of her presentation is Breaking the Wall of In Situ Tissue Engineering.
Hi everyone. These pictures are familiar for all football fans and footballists. Yes, I'm talking about the muscle injury. Intensive injury and severe damage normally can't be repaired by the muscle itself. So the available treatment is tissue transplantation, which is difficult because of the rejection of the transplanted tissue and also finding the suitable donor. So the injured person actually suffer from the loss of the functional muscle tissue and restoration taking time, and also there is expensive healthcare cost. So the alternative treatment is tissue engineering, which is right now saving the healthcare cost in US for $250 billion per year. But what does it mean? It means we take the cells from the same patient and expand it in laboratory on the suitable substrate, and then we, on the proper condition, we make the functional muscle construct and transfer it again to the body. But the big challenge is here that making this functional muscle is difficult, and we need again surgery, which is painful and complex, to return it back to the body. So the wall is right here that we have to break by in situ tissue engineering. And how? We're going to make a shortcut, make this complex process simpler and faster by removing this complex part and transferring the cells directly to the body. So what is my solution? Our solution is using the local delivery of the cells, which are attached to the carriers, and injected to the injured tissue, and after a while the cells can regenerate the damaged part of the tissue. And we design the polymeric carrier from the cheap commercial polymer, polylactic glycolic acid. We load them by cells, and then they look like Italian spaghetti. The polymeric part is like a spaghetti, and like these green dots on the spaghetti are like cells, which are attached to this carrier. And as a proof of concept, you see here the microscopic picture of this carrier with the cells attached to them. And easily, with the syringe needle, we can inject them to the injured tissue, and after a while, this polymeric part is degraded, and cells regenerate the damaged part of the tissue. So here, also, you see the injectability. These carriers are flexible, and there is a high cell survival rate than direct injection of the cells because they mechanically support the cells and make an environment like a body for them. And I believe this idea of the cell-loaded nanocarrier can break the wall of in situ engineering, and it's not only for the muscle tissue, it can also use for the heart muscle. And that can improve the health and life quality of the million people worldwide.
Okay.
Thank you.
Open for questions.